After we heard a very interesting talk by Ralph, in which he gave both the bright sides and vision as well as some of the obstacles in drug development, we will hear now few examples of the excellent research done in the university in the field of chemistry, in the interface between chemistry and medicine, and in the field of computational computer-assisted drug discovery. And the first will be Professor Doron Shabat from the School of Chemistry. As many of you know, Professor Shabat, Doron, is a very innovative and creative chemist full of new ideas, and today he will give us a glimpse of the different new system that he had developed over the years and maybe also future plans. Doron, please.
Thank you, Udi, for letting me speak in this symposium. Let me just start the presentation. Okay. Anyway, today I would like, as Udi said, to give you a concept from a point of view of a chemist or a bioorganic chemist. I will probably not show you any molecules today, but I will show you cartoons that represents a molecule that we are able to translate this cartoon to real molecules. So let's start with maybe the most simple drug delivery system that exists, which is a prodrug. From terms of from a point of view of a chemist, a prodrug is a drug molecule that has been modified, usually by what we call a protecting group. And if you remove this protecting group, you actually release this, the active moiety, the parent drug. And in some cases, the protecting group could be related to a specific enzyme. And therefore, it could be an enzymatic substrate. And if the prodrug is activated at a certain organ by a specific enzyme that is overexpressed at that tissue, then this can be this kind of product can be targeted or delivered to this specific or certain tissue. Now, in the classic prodrug, each cleavage of the enzyme substrate will release one drug molecule. And we are asking ourselves if we can release more molecules of drug by this single cleavage event. And this could be relevant if you are limited by the activity of the enzyme in the targeted organ or tissue. For example, how would we release two drugs of molecule if we, by a single cleavage? So the graphical solution to what I just mentioned is something like that. So you have a third moiety here, which we call it a chemical adapter, and you link two drugs to it and one enzymatic substrate. And the way it's going to work is that once the system is stable as long as the enzyme substrate is on it, but once you cleave it, this adapter has a unique reactivity. It can translate this bond cleavage into two additional bonds cleavage of that connect the drugs to the system and overall release these two drugs. So by this way, you can actually amplify your release. And if you can design and prepare a chemical moiety that will give you this amplified release, you can further extend it to what we call dendritic molecule. And you can build a tree-like molecule, and it's going to work the same. You cleave once, the head of the adapter is free, the two tail will be cleaved, and now additional cleavage will give you the release of all these four molecules. So based on this cartoon, we develop our self-immolative dendrimers. We use the phrase self-immolative. We borrow it from a different topic in chemistry, but the idea is that the linker actually sacrifices itself in order to implement its function, and the function is the amplification of the drug release. So this is one system that we developed and tested so far in vitro. We didn't reach yet in vivo with this system, and there are several limitations to this system, but there are also several advantages. For example, you can think about the most simple system and have two drugs, but two different drug moiety, and if these drugs work in synergism, then you can achieve the release of these two drugs simultaneously at the same location by this kind of platform. So this is one platform that I'm presenting today, and the other one is related to a polymeric structure. So similarly to what I showed you before, if we can design a polymeric molecule that has a head trigger, but once you cleave the head trigger, you can initiate a head-to-tail disassembly, domino-like disassembly, that overall release all these monomeric molecule. And if you think on a diagnostic application, if this monomer give you a signal, then you actually generate a lot of signaling molecules by a single cleavage event of this head trigger. The advantage in terms of synthesis here is that you can take an appropriate monomer, submit it to polymerization condition, and then cap the trigger at the head of the polymer in the last stage, and in this way you will be able to prepare a large molecule that contain all your monomeric molecules. So the application, one of the applications that we are evaluated with this kind of molecule is sensing, and if the monomer is fluorescence when it's free, but it is quenched when it is within the polymeric chain, then you can achieve the amplification of the signal that is related to the analyte that you want to identify. And the way it's going to work here is that this monomer are going to, through a sequential disassembly reaction, it's based on a unique reactivity that's called in chemistry quinone-metide elimination. And those quinone-metide species, if you design it properly, after reaction with a water molecule, can produce a fluorescence molecule, and that's how you can actually amplify fluorescence. And this cartoon, this animation cartoon is showing, demonstrating the idea. You cleave the substance with the analyte, and then the polymer is being broken to its monomeric unit. After reaction with a water molecule, you generate a fluorescence molecule, and you can actually amplify the signal that is initiated by the analyte that cleave the initial trigger moiety of this platform. So we have demonstrated this concept by a polymeric molecule. This is the structure, and here we use BSA as the analyte that we want to identify. And BSA can have a specific chemical reactivity to cleave this red moiety. It's not really important why, but overall you can measure the fluorescence that is amplified by this molecule upon reaction with the BSA. I've shown you actually those two systems now, the self-immolative dendrimers and the self-immolative polymers. And obviously, we look at those platform as amplification moieties. Both the polymer or the dendrimer can amplify a cleavage event into multiple release of small molecules. So I want to show you now a parallel approach to that is based on the dendritic-like structure. If we synthesize high generation of self-immolative dendrimers, it is a tedious stepwise synthesis that accompanied with a lot of technical issues. And we wanted to achieve the self-immolative amplification, but with a very simple molecule. And we took the most simple immolative dendron that can exist, call it an AB2. It has one trigger and two leaving group that will release here. We call them now reagents. You will immediately understand why. And the idea is that these reagents are inert. They don't do anything as long as they are connected to the platform. but by a reaction with a specific analyte that you want to identify with the head trigger, those two reagents will be released from the platform. When they are free, they somehow can acquire the chemical reactivity of the analyte of interest. If this is true, if this will occur, then these two reagents now will react with two additional dendron presented in solution. And therefore, we'll be able to cleave two additional triggers, release these four reagents that will activate additional 4 dendrons. And therefore, you will get an exponential process that will release all these reagents. And we call this concept dendritic chain reaction and auto-amplification exponential system. All we need to do is to follow this kind of reaction is to adjust somewhere a reporter moiety, a moiety, a small molecule that will give us an indication to every cycle that is occurring. So we redesign our system with the two reagents, and we added what we call a reporter molecule. Upon cleavage of the trigger by the analyte of interest, the two reagents will be released, but also the reported that will that we can read it spectroscopically, and in this way identify the disassembly of the system. And as I showed you before, when the reagents are released, they will activate 2 additional dendron and so on. And that's the dendritic chain reaction, auto-amplification. And here, this is the amplification cycle that overall releases all reporter molecule in an exponential manner. And again, we have a small, short animation movie that demonstrate this concept. And the idea that you put your analyte of interest to the solution, it reacts with the probe, release the signaling moiety and 2 other reagents that activate 2 dendrons. In every cycle, you double the amount of dendritic molecule that are being disassembled through an exponential pattern to overall release all to break down all the molecule present in the solution and therefore to generate a strong diagnostic signal. And we have demonstrated, as you can see, this is a cartoon, and of course, if you can find a different chemistry that will fit to this cartoon. And we found at least 3 different chemistries that fit to this cartoon, and we have shown how we can amplify a signal in order to identify a specific analyte in a diagnostic assay. The last topic that I want to present to you today may be more related to the center, to our drug discovery center. And this is current research that are being done today in my lab together with Ronit, who you will hear later after my talk what Ronit have to say. But this is a classic collaboration between chemistry and biology that enable us to maybe develop new drugs already in the stage of academic research. And let me show you what I mean by doing this. My problem is related to a general problem in drug delivery. Think about a bioactive molecule, a small molecule that has a wonderful cytotoxicity or biological activity, and you really want to use it in a drug delivery system like an antibody drug conjugate. The problem that you cannot do it is technical. It's technical because your wonderful small molecule has no functional group that you can link between the small molecule and your targeting carrier through a linker. You simply don't have the option from a chemistry point of view. It doesn't have the chemical reactivity or the chemical functional group that allowed it. And we wanted to address this issue because there are several molecules like this that are known in the pharmaceutical science, and also some that have really only limited conjugation option, and we wanted to actually address all this issue. And our solution or approach to solution of this problem is simple. How about simply develop a chemistry, kind of a general chemistry, that will introduce a functional group on your small molecule, small bioactive molecule. If you have the functional group on it, then it's no problem to connect between your targeting carrier and the drug through a certain controlled release known linkers. But this can only work if your new moiety here will keep or maintain the biological activity of the parent drug. Of course, you generate here a new moiety. It's no longer the same molecule that was bioactive, that was a drug before. You generate a new drug. But if you do it in a specific way and modify a region in the small molecule that will not interfere with the binding to the receptor or in general in the biological activity, then this can be achieved. And indeed, that's what we were doing. This is going to be the only slide today that I'm showing you molecules. But I still don't, I mean, these are relatively new results in the lab. This is the reagent that we develop. It is based on chemistry that developed by a collaborator of mine from the Scripps Research Institute, Phil Barron from Scripps in San Diego, La Jolla. And what we have done here is develop a chemistry that allows CH activation of a molecule like campthotecoin. And the functional group that we introduce is a ketone. Ketone can be used for a drug delivery system through a couple of approaches, either acid labile release linkers or photolabile release linker. And we had a hunch that with ketone it can work because it has what I call a relatively innocent behavior. Innocent behavior, this is my definition, means that it will not interfere too much with the parent activity of the original drug. And indeed, when we check the cytotoxicity of the ketone analog versus the parent drug, and this was done here in the lab of Ronit, we found out that we virtually have identical cytotoxicity. And I'm showing you only one slide, but this assay was repeated in about 10 different cell lines. And in all of them, the cytotoxicity of the ketone analog was almost identical to the cytotoxicity of the parent drug. Now, yes, of course. I didn't get it. Did it stay as a ketone when you were testing it? Did it stay as a ketone? Because I mean, I'm not quite sure that I'm understanding.
You test the cytotoxicity.
Yeah, of the ketone. Yeah.
So I'm saying, does it remain a ketone, or does it react with compounds that are in the assay system?
Okay, so I believe that it did stay as a ketone, and maybe I don't have an absolute proof for that yet, but when I described this linker before and I say that we chose a ketone, and I use the phrase innocent reactivity, and although a ketone is an electrophile, it's not a strong electrophile, and my hunch that it did not react with any other molecule in the assay. Anyway, this is one example. We repeated this chemistry on 4 different drugs. 3 out of the 4 we were able to observe identical reactivity. In one of them, methotrexate, for example, we completely lost reactivity. So it's not, I cannot say that this is an approach that will work on any drug. Of course, it has to be a very specific one that is suitable for this kind of chemistry, but 3 out of 4, I think it's a good start. And just a gimmick that came out of that, Aldrich has decided also to commercialize this compound. And if anyone want today to try this chemistry, they don't have to prepare this reagent, and they can buy it directly. It's always, if you can have, yeah, well, this is not, it's not a straightforward chemistry. I mean, it's not like you mix the, you take usually excess of the reagent, 2, 3, or 4 equivalents even of the reagent versus 1 equivalent of your drug. And it will not, I didn't describe all the chemistry. It has to be a certain CH bond that can react with it. It's usually the most negatively poor CH electron bond. And of course, it can be fitted only to certain kind of molecules. Yeah, but with this slide, actually, I'm concluding my talk. I've shown you today our approach to amplification and drug delivery system. And also, in the end, I show you how we can actually prepare by a simple modification a new drug analogs that present advantages if you want to use them in bioconjugation. This is one of the reunions of the group that we had 2 years ago. I'm showing this large group of students that represent maybe 4 different groups of mine over the last 15 years. And one of the best things in Tel Aviv University, there are 2 main advantages in Tel Aviv University. One of them is that it is inside Tel Aviv. That's a very important one. But the second important one is that because we're in the center, we are exposed to, I think, the best student in Israel. And we have the privilege of working with wonderful, talented young student. And I think I owe this presentation to them today. And although I don't see her name here, I want to thank Ronit that will speak after me. Most of the work that I presented here was done in collaboration with students of her group. And thank you, Udi, again for everything.
Thank you very much. I'm the one who has to keep the time for a few questions, but I have to ask you one question. Is the chain reaction could potentially be suitable for detecting a single molecule?
Potentially, you see.
So with the kinetics of how much time will it take to amplify?
Yeah. So we are still far from there, okay. And the reason is because there is always background reactivity. Okay, this is the second. And if we'll be able somehow to get rid of the background and speed up the reactivity, the process reactivity, then we can identify lower concentration, of course. But we are still far from there. Life are not as simple as you saw in the movie. Yes.
Any burning questions? Thank you very much.
Okay.